specifically for the latest data types, especially long reads and high C, to produce like chromosome-scale haplotype-resolved genomes. So let's get started. So here is the outline of my talk. I'll present first the reference-based phasing. What's a phasing is, and how can we perform phasing in the space of a reference genome. For example, in the humans, when we already know the reference genome. And then I will present a generalized de novo method in the cases when genomes are highly divergent from each other and we cannot produce phasing directly with the reference. And further, I'll present some of the applications of these methods in the medical genome method, specifically cancer, and also some of clinical relevance. So we can keep this lecture interactive. So if you have any questions, feel free. Okay. So first, what is phasing or haplotyping? Imagine we know these two variants and their genotypes, which is AT and CT. And what we actually want is the haplotypes, which means A is connected to T and T is connected to C. And the process of obtaining these haplotypes is known as phasing or haplotyping. So, there are many applications. These haplotypes are helpful to study human migration and evolutionary selection and population structures, and also to understand the causes of diseases. So, over the past few decades, there have been huge advancements in the sequencing technologies. For example, starting with the second generation sequencing datasets, where the read lengths are about 250 base pairs and the reads are very accurate. And to the more recent ones, third generation sequencing, such as PacBio and ONT. So the PacBio and ONT traditionally have the read lengths of about a few tens of kilobase pairs, and the error rates were pretty high, like 10 to 15%. But in the last 2 years, there have been a major breakthrough in the PacBio Hi-Fi specifically, where you can get high accurate reads, about 99% accuracy. And also, Nanopore has also made advances in the difference mode, so that accuracy has reached towards 97%. And then there are more long-range technologies, which are single-cell based, for example, strand-seq, and also high-C, which actually captures the spatial structure of the chromosome, which gives us these long-range information. But still, the underlying data is still short-read sequencing. So the next question is, how can we utilize these new advances in technologies and data types to perform the phasing directly from the reads, and we say that as read-based phasing. So the advantages of read-based phasing is you can phase de novo or rare variants, and you also can produce chromosome-scale haplotypes. So more traditionally, techniques like genetic and statistical phasing have been followed, like for example in 1000 Genome Project. So in genetic haplotyping, essentially you make use of prior information and try to explain the haplotypes based on the retained loss of inheritance. So the advantage of genetic haplotyping is, for example, you have a variant if it is heterozygous in all the individuals, you cannot obtain phasing for those variants. And then the next approach is statistical phasing. Here the underlying idea is the haplotypes are inferred from the genotypes based on the evolutionary model that lies in the population patterns. So you try to explain the genotypes, haplotypes based on this common ancestral information that is consistent with the target genotypes. So let's see. So in this plot, on the x-axis, you see the number of samples in the reference panel, and the y-axis is the phasing derivatives. So if you use about thousands of samples, the switch error rates are about 1.5%, but if you increase the sample sizes to about 10K, 100K, then the switch error rates reduces to less than 0.5%. So this suggests you require large sample sizes if you only want to achieve high quality haplotypes, which are sometimes not available in some combinations. And another limitation is you cannot phase de novo or variance through statistical phasing, and also you cannot get the chromosome haplotypes in this. And also another important medically relevant regions are HLA and here, so we cannot phase these regions by using statistical phasing. So to overcome these limitations, here we follow a read-based phasing for single regions, which is defined as follows. So you have a reference genome, and you know the SNPs, the genotypes, and you bring in the reads from target individual. It could be long reads, it could be IC, it could be short reads. So, and then you map these reads to the reference genome. So the alleles that span these SNPs to each read is encoded in red. So, and now the goal of read-based phasing is to partition the reads to two sets, because we are working right now on normal human diploid samples, and so that's why there are two partitions. So, in the case when there are no sequencing errors in the data, this problem is trivial to solve because you can easily make this partition such that the alleles between any two rows match. For example, this and then the main cluster. And then you can bring another. But in practice, you get a lot of sequencing errors, and the goal here is to detect these errors and to flip them to get the final conclusion. And to solve this problem, we followed a minimum error correction problem or a weighted minimum error correction problem, which is you flip the minimum set of entries such that conflicting partition exists, and this property holds if the alleles between any two rows match. For example, if we change this G to T and this G to P, T to G, sorry, and then we can make these partitions and get the final outcomes. So this was the basic formalization of the problem. So now the next question we ask is how can we get dense and whole chromosome haplotypes of different genomes when we have a reference genome. So imagine we have a reference genome and we align the reads from different technologies to the reference genome. So first of all, we use a strand-seq technology. That is, the beauty of this technology is that you have a global information, meaning so you have this series of ones and then some gaps and some series of ones, but this each row comes from one haplotype. So this is the information you get from this data type. But this is a single cell technology, so that's why there's a large sparseness in the data. But to circumvent this issue, we bring in the dense information from the long read sequencing data. So we have these series of different ones, but this information covers a few tens or 15 SNPs, but it's not on the whole chromosome scale. So next, the question is how can we solve these instances that include some sparse information and also this dense information in an optimal manner to produce the whole chromosome haplotypes. To solve that, I implemented a dynamic programming-based algorithm, which actually solves these map instances in WhatsApp, and gives optimal solution and can phase human genome in about an hour or so with a standard program. So then we were interested to do some experiments on the haplotype quality by combining different technologies. So here I present the results on the human chromosome 1. So when we combine PacBio and 10x, we observe the following. So each panel represents the different some sample images from the background data, and each color is a phasing sign. So more the colors are, more fragmented is phasing. And then there are centromeres which we cannot phase yet. So the ideal case would be if we see one color through the whole chromosome. So that's our goal. So when we combine all the PEC-bio and 10X, we still observe a lot of colors, meaning the phasing is still fragmented. But when we combine strand-seq with 10X, we for the first time produce this chromosome-like phasing. But there are the centromeres that we cannot phase. And we also looked into the phasing quality by comparing against the ground truth that is produced from genome model, because they use the trio information to produce these haplotypes. And we observed that we could produce less than 0.5% of switch error and about 1% of so this study shows us that by combining long range and long reads, in the space of the reference genome, we can now produce chromosome-scale haplotypes, but excluding those centromeric regions. But in this study, if you observe closely, there is a reference bias involved. Imagine you want to bring in the new populations like African. In those cases, this study can fail. So in order to address that, we actually proposed a new graph-based method, which is to produce a diploid assembly for the trios or in general. So in this study, what we do, we combine short reads and the long reads to produce the chromosome haplotypes. So first of all, we use standard like spades to produce this graph, which is actually, I will walk you through. It's actually a concave graph by using the short reads. Since short reads cannot span a lot of repetitive regions, so we don't yet study those complex regions. So the aim is to get a series of bubbles and phase these bubble chains. And so we observe the graphs like this. So you see these special structures in the graph. Those are known as bubbles, and which has a common start and the end node, and these represents the haplotype variations. And there may be some complex nested variation, for example, a SNP inside SV, so that also forms like a multi-allelic bubble. So the next, we detect these bubbles to find the areas of heterozygous variation, and we also find these bubble chains in these graphs. And then we align the long reads through the graph by using seed and chain algorithm. And next, the goal is to phase these bubble chains to produce the diploid assembly for the trios. And to solve that, we generalized this map framework that I introduced earlier to actually graph version of pedigree map. So which is defined as follows. So imagine you are given these bubbles and these alleles in the bubble, and you are given the alignments through the graph. And now the goal is you want to find out, essentially for one pedigree, you want to find out 4 partitions because of the recombination event. So you have 2 haplotypes from mom, 2 haplotypes from dad, and the child is essentially a mosaic of the haplotype of father and the mother. So that's why. So we want to flip the entries allowing minimum recombination events such that 4 partitions exist. And the output we want is diploid assemblies for every mutual entry. So, and we solved this again in a dynamic programming based method, which is based on, again, the WhatsApp algorithm. So essentially, in that connected component, we first find the optimal path pair for m bubbles, and then we extend it to n plus 1 bubbles. So at any stage in a bubble, it's conditioned just on the previous k minus 1 or if it's m plus 1 bubble, it will be conditioned on the first m bubbles. And then we iterate until the last, and then we back trace to get the final haplotypes. So at every step, we remember the possibilities only from the previous step. So this helps to make this algorithm linear in the number of bubbles. And we continue until the last bubble, and then back trace to get the optimum paths corresponding to this global optimum. So I'll walk you, for simplification, I'll walk you through an example which is for a single individual, and then we can easily extend for the pedigree version. So imagine you have a bubble, and you have these reads R1 and R2 aligned. And now, in the first bubble, you would like to find out for every path pair, so you start to consider all the path pairs that the reads align to, and we compute this bipartition score. What's the minimum score to assign the reads to the bipartition? So, for example, here the possibilities of bipartitions are, there are 3 reads, you assign all to 1, and then other possibilities are so on. So it's essentially 2 power the number of reads minus 1, so the number of possibilities of the bipartition. And we calculate the score as follows. For example, we are on B1 bubble, and we want to calculate the score for this R1, R2, R3. And then the one as you put, so imagine the Fred score for each of them is 10. Then either you put all of them into this path, or you put this one to this one. So it's 10 or 20. And we calculate similarly the cost score for all the bipartitions and for all possible orderings of alleles. And then when we go to the next bubble,
what was B1?
Sorry?
B1?
B1 is a bubble, like first bubble.
Okay, the bubble.
Yeah. And then we go to the second bubble, like B2. And then here, we again compute for every path pair out of 3 paths. So we calculate the same, but we given the cost for all bipartitions for the intersecting reads. So this is the main point here, that we consider the bipartitions that are consistent across 2 bubbles. So that covers first and the second bubble. So similarly, we start to calculate for all the bipartitions. For example, here this R1 goes there, and R5, and there is R7 and R6. So we start calculating all the partitions, but for the recurrence cost, we minimize over all the path pairs. For example, here there is R1 read that goes and R5, and we will take the minimum of these intersecting reads that covers first and second. In this way, we iterate over until the end of the bubble chain, and then calculate the paths. So for the pedigree version, right now we are doing for the single individual case, but for the pedigree version, we do similar score in a joint fashion for all 4 partitions. So we perform some, because this was based on, the graph was based on short reads and the long reads were threaded. So we started with small chromosomes, and we simulated different heterozygosity rates ranging from 0.5% to 1.5%, and we took different coverages of Banbar. And here we benchmarked these assemblies against Trio-Kanu. That was the state of the art that time. And we observed that we could produce higher quality assemblies relative to Trio-Kanu, and it was 89%, we could produce 99.3%, and also the continuity was higher because we could partition reads better than the Trio-Kanu. So, and then we performed experiment on the real data for chromosome 22 only, and this suggests that we can generate high quality diploid assembly even at 50x long coverage for So, as we were working through this workflow, in the meantime, there were some major advancements in the sequencing technology, that's the bio-Hi-Fi. So that's why we now started to work on the whole human chromosome, or whole human genome, moving towards the chromosome. So here we made use of these newer data sets like Hi-Fi, which now has these read lengths have reached to 25 kilobase pairs, and it gives us a very high quality reads, which is Q20. And also we made use of the Hi-C technology. So Hi-C gives us the long range information. So the next is how can we combine these newer data types, Hi-Fi and Hi-C. This is like just a last year work to produce these diploid assemblies. So a little bit background on Hi-Fi sequencing. So essentially you need here a high molecular weight DNA, which is easy to get for these cell lines. And then you ligate the adapters, and which lead these primers to be annealed, and the DNA is polymerized. And then DNA gets circularized, and then you essentially perform repeated passes of sequencing and then call the consensus over this set of reads that you get, and this produces very high quality reads, and on an average of 20 to 25 kilobases. And still these Hi-Fi reads are not long enough to span the repetitive regions like centromeres or rDNAs. So to stitch these pieces together, we made use of Hi-C technology. Hi-C actually captures these spatially close segments. For example, this is the whole one chromosome, and then we digest and ligate the adapters here. So this means this and this point of the genome sequence that occurs on the same chromosome. And this has been very helpful to stitch these pieces. So without going in much detail of the technology, but I would present a more take-home message that is happening in this data. So on the x-axis, you see the genomic loci, and on the y-axis is the contact frequency. So darker the color is, higher is the contact between these genomic loci. So up to 10 megabases, you would observe a higher contact frequency, whereas if you go further, it starts to get lower. So up to 10 megabases, this signal that is to stitch together these sequences on the chromosome haplotypes is very strong. But later on, it can be even different chromosomes and so on. There can be many errors in the data. So next to now put these data sets together, we come on the computational end. As we know, this as Julian mentioned that genome assembly started with the human genome project when the reads were short, and the graphs look like this, so messy because 50% of the human genome is short pattern repeats and also high order repeats. So I hope, like most of you are familiar with these, how do you construct graphs like this, so I will not go in detail. Or if we want, we can go in detail of how to construct these divergent and overlap graphs. So how many of you knows about divergent overlap graph? So yeah, I'll briefly give an overview. So imagine this is the genome, right? And A, B, and C, these are the unique regions of the genome. And there are few repetitive regions like this R. So the reads, if the reads are not long enough, then we cannot map predict whether the read is coming from, for example, this R read, we don't know where it's coming, whether it's coming from this read or this or this. So now to construct these, we take the reads, and in the overlap graph, each node is a read and the connections are the overlaps between the reads. So because of these repetitive regions, you start to see these tangles in the graph. And in the divergent graph, what you do, you break the read into these fixed length words, and now each node is that word and the connections are the overlaps, and it's typically a bidirectional. And now the goal, if we want to get the haplotypes, if we want to find these haplotype maps through the graph. So when we constructed this graph from short reads since 2 decades, the graphs were very tangled because of the repetitive regions. But when we moved from short reads to long reads, now we can span some of these regions and we can find these unique overlaps and find the orderings of the reads. So this produced better graphs, and the assemblies started to become a bit more continuous. But still, the major issue was that both the haplotypes, they were collapsed into one consensus sequence. So even at this stage, even if after 2 decades we have finished the reference genome, and still the reference genome doesn't contain the true nature of the humans, that is the diploid nature. So there were some advances to produce the diploid haplotypes. For example, Falconansia, trio Kanu. This uses trio information, and it was designed for PacBio CLR reads. So it was the haplotypes that it got were not on the chromosome scale. There were many, as we see, there are many colors here. That is the fragmented. So to overcome this issue, here we proposed a new idea that actually works specifically for the hi-fi reads, which is to use the phasing information directly in the read overlaps. So what we do, we find read overlaps, but within the overlaps, we find these heterozygous SNPs. And if the alleles match in over these SNPs, we say these reads are coming from the same haplotype, and these gray and gray ones, these are coming from the different. And we import this information in the graph. So here, each node is a read, but the connections are now the haplotype specific overlaps. And then we see these beautiful structures in the bubbles in the graph. And this idea we demonstrated especially for the segmental duplications, which are highly identical repeat units, and it seemed to work fine for those segmental duplications, and we could produce continuous assemblies inside. But this idea was then implemented by hi-fi-SM on the whole genome level. And here we propose a workflow that combines hi-fi and hi-C to produce the chromosome-scale haplotypes. So we take the hi-fi reads, and first we produce these contiguous sequences, and then we scaffold them using hi-C. And further, we remap these hi-fi reads to find the heterozygous SNPs, and then partition these reads to 2 sets to get the 2 haplotypes. And then we were interested to see the runtime, and we, for the first time, could produce these chromosome-scale haplotype-resolved assemblies within a day, which used to previously take about a few weeks. These were due to the advances in the technology and also the new algorithms and workflows. So then we applied this to public genomes like H002 because there's a lot of data available for benchmarking and also a lot of ground truth available. So here we observed that we can produce about 3 gigabases of each haplotype, which is as expected. And we can produce better continuous assemblies than the tree of Kanu, and we can phase a larger fraction of the genome, about 8 times better, and the switch error rates are comparable to tree of Kanu. Then we also applied to other genomes, and we observed a similar trend. So Yeah, and then I don't know what's the time. Shall we take a break or? Okay. Yeah, I think maybe let's take a break for 10-15 minutes and then we can resume. So it's in that
we use
So we even we can do one partition like alignment. It's a very expensive set.
then I understand that. Basically, it's what's part-time problem, which is the case that you need to distinguish between tasks. You want to start it for choose
Like this,
and it's computationally interesting.
a lot. Yeah. It takes 2 days now.
They use it for the genome or okay, that's good.
Yeah.
But if you write everything and faster,
I think it can be done like in half a day. Half or maybe 12 plus 3-4 hours, like 15 hours. With standard computer.
so which means that you have the sound geographies that are in distance of design.
Yeah. If you are interested in this topic, we can.
The business area was trying to, I was already voted once about the map on a graph. But then I do on the internet or something.
So I really wanted to work on this problem, but my last year ongoing engagement.
Sorry?
My last year ongoing engagement.
I'm wanting to try to figure out maybe fast.
I think I have some ideas.
But this is you're not too devoted.
No, it's not my
I see. Because really, nobody wants to do themselves. Okay,
and it's not essentially just like data.
okay, I think that it could be interesting. Because we did
and we had both matched and tumor for this. So we sequenced both of them to to about coverages of 30 to 40x. And then we assembled these cancer genome to using our algorithm, and then we mapped these genomes to the reference genome to call the structure variations. And here we observed about 26,000 somatic plus germline structure variations, out of which there were about 13,000 insertion, 9,000 deletion, a few tens of duplications, and also a few tens of inversions. And we observed and we also compared these structure variation calls against the standard short read sequencing based calls, and we observed that about half of them were missing in the short read columns. And then we were interested in specifically the repetitive regions of the genome, because that's where these long reads have an advantage over these short read based technologies. So we considered different repetitive classes. On the x-axis you see ALUs and these ERBs, L1s, L2s, and sign elements, and these telomeric stuff, and so on. And on the y-axis are the number of variants that are consistent across different methods. So we took different call sets. So there is one somatic variant call set from New York Genome Center that was produced using short reads, and then there is one from Netherlands, and there is single cell base sequencing from USC guys, and then we performed this all versus all across all this performed essentially 4-way handshake. So as we observe that this blue bar represents the consistency of our method with the single cell based method, and which seems reasonable, because the single cell, they are targeting the cancer clones, and they are able to find out the variations that is happening in each clone. So this suggests that our long read and IC based method is at par with the single cell based method. And single cell also is expensive, so at this stage it's unclear which is better, but both are delivering as good results as they are. And then we also looked into this P10 deletion, which is about 12 kilobase pairs, which is actually typically missed in the short read based calls, but we can find out using these long reads stuff. And then we looked at some of these clinically relevant regions. For example, HLA. HLA is a highly divergent region. It's different in each even normal genome, and also it has known to have associations in many psychiatric and autoimmune diseases. So HLA-A has been unknown for a long time because of its complexity, because it has a lot of, especially these DRB, DQ8, DQB, they have short tandem repeats that are repeating many times. So we took our assemblies and we aligned them to the reference genome. So darker this color is, higher is the divergence of each genome to the reference genome. So you will observe that in this DQB, DQA, there is a divergence of even 10% to the reference genome. And then if we study them using reference base, we will typically miss all this variation that is to be unique to a genome and also to the disease. And we also observe the variation between the haplotypes of the same sample. So you see in H1 there is a structure variation here, but in H2 is not. And similar pattern we also observed in KIR regions. KIR are another killer cell immunoglobulin-like receptor region, which has associations with autoimmune diseases, and we observe these 10% there were in these regions like KIR, DL1, studies suggest that this Hi-Fi and Hi-C based sequencing can also go into through these complex clinically relevant regions. Next, I will talk about our recent prospective article that we wrote. Like now, how can, so one is you study each genome separately, and another is you go beyond one genome, like to enable this new era of pangenomics. So chromosome-scale haplotype-resolved pangenomics. So which is defined as follows. So you have a set of representative sequences of chromosome copies within species populations. For example, you have populations of Africa, Europe, and so on, and then you map these to the reference genome, and then you put this whole variation from different populations in the form of a pangenome. So essentially, you can also import the tandem repeats, for example, in the form of this bubble that can happen in different individuals and populations, and also the complex assays. So the pangenomes can enable comprehensive assay discovery and also the long-range interactions of the haplotype. For example, there may be a variant is interacting with this variant, and also the functional relationships. And furthermore, beyond human populations, this pangenomics could be helpful in identifying superior haplotypes associated with stress tolerance or disease risk, and also to enable the new cultivars for food security and food production. For example, imagine you have this rice genus and they have these different types of species which are either cultivars or white rice. So, and then there these hybrid crossing that may enable these new cultivars, and if you cross this the new haplotype with improved stress tolerance. So traditionally the pangenomics have been defined as follows. So you are given the chromosomes and the haplotypes, but they are collapsed into a consensus sequence, and also they are not on the chromosome scale. So there are typically some gaps in the consensus sequences, and they're essentially a collection of the collapse and gap haplotype sequences of multiple genomes. So there have been some improvements, some advances in the computational methods in the graph pangenomics, as you guys have already just to give a recap. So one of the first methods was in the graph pangenome was you take a reference genome and then you take variants in the reads and put these variants into the reference genome just one by one, left to right, and you get a graph, so which has the variants from different individuals in the populations, and it's a DAG, so it has a very nice structure, so very easy. But now with these new advances like, for example, HPRC or tomato reference pangenome and many others, you start to get these chromosome-scale haplotype sequences. And there have been few methods that directly work on the assemblies without using the reference genome, like PGGB. So one of the method is K1Ts from Stanford. So essentially, they break these assemblies or PCFs into the K-mers and start to put the K-mers into the reference. And then another one is the mini graph on the progressive cactus. So the mini graph works iteratively. It starts with one reference and then find the structure variation in the other sample and adds and keep on adding incrementally. But mini graph alone cannot do the base level alignment. So to circumvent that base level, the community generally uses this progressive cactus that can perform the base level alignment of these structures. So and the next one, the most recent method that can do the base level alignment is a wavefront algorithm that I believe Willie and others have already covered the details of this. So this can these methods can again produce a graph with pan-genome, but there can be cycles in the graph, but with some greedy heuristics, generally the output produced is a DAG. So, but these methods, they have some limitations, like they can scale to few tens to hundreds of genomes, but may not scale it to thousands. So here in this article, we propose a new model, which is a hierarchical model that allows the biologists to visualize the variations at different levels. For example, you are given this phylogenetic tree, like class, genuses, and species, and you have these representative sequences. So here we take, it's essentially a variation graph. Here each node is a subsequence, and then the connections are the adjacencies between the subsequences. But there are two types of connections here. One is the inter-level, which is intra-level, which is at the same taxonomy level, and another one is this inter-level, which actually gives the relationship across these different levels. And the relationships is generally kept through either through these similar sequences that can happen across species populations, or it can be done in the space of the reference genomes. So we are still working on this. That is still under development. If you guys are interested, So yeah. So this brings me to the end of my talk. So here first we studied the reference-based phasing, like how the map framework works to produce a chromosome-scale haplotypes. Then to overcome the limitations of the reference bias, we studied the graph version of map and also how we can extend the graph version to a pedigree version, and some latest advances in the combination of high-phi and high-C to produce chromosome. And finally, I presented some of the applications of these algorithms to cancer genomes or the medically, clinically relevant features. So in a nutshell, we are excited about new algorithms and data-driven approaches to combine massive erroneous prone data, which is generally hard to integrate, and we want to yield meaningful insights to enable precision medicine and understanding of diseases using haplotype-aware strategies. So I thank my group, my mentors, funding agencies, and collaborators. Thank you.
Questions? I have one. Okay. Please go.
Yeah, I have two questions. The first one is, what biological assumptions do you need to take into consideration when you try to detect haplotypes? So my question is because recently I understood that if you want to detect non-allelic haplotypes, you need a common region between these two. But in this case, I think that we don't need that. And my question is related to how you approach the recombination.
Yeah. So the first point is we have designed like the initial algorithm for the diploid case, like the MET thing that we have designed that is actually right now works for the diploid case. So there are some limitations for the polyploid. actually we could find out the recombination events. Like the algorithm can find it. But we haven't validated those, like how, because there could be one layer and there could be full switch. We haven't validated those recombination events. So that's a good point of further investment.
Yeah, the other question is, in your pipeline, I noticed that you mentioned when you combine short and long reads, in what step you use deep variant?
You use?
Deep variant?
So we haven't used deep variants. So in this, in the variant calling step, yes, that's right. Yeah.
That uses only short reads?
So it's a hi-fi reads. So we use hi-fi and hi-C here, and we call variants here using deep variant.
But there you put sleep maybe.
Yeah. Snips.
Not with
No.
Okay. Other questions?
What usually ask the speaker to, if there are young students, give your insights about what's going to expect them in the future.
Like from the algorithmic, I think there is still a need of like new computation that can enable routine, because still these workflows take like a day. It can be really done in a couple of hours. So that could be a good algorithmic challenge. And in the future, probably these data types will keep on improving, and then also the algorithms will need to improve. And also, right now we are taking this to a more like disease-oriented, like in rare diseases or cancer, and other types of
So I have a question related to cancer, and not only. In cancer, we have that the number of haplotypes increase.
Yeah.
Okay, so we're not doing the legal mistakes.
Yeah.
How do you face this issue?
So when we the hi-fi reads on a reference read, right? We find the reads that are similar to this, and then for every read we just find the reset. So this whole infrastructure enables plot diagnostics. So it can, but the column 8 to 9, it's not too complex. It has just a plotty of like 3 or 4.
So it's limited in general to 3 or 4.
I mean, it has been, like in theory, there is no limit, but it has been tested only for 3 or 4.
Okay.
Like in theory, it's plot diagnostics, but
Maybe for the state.
Exactly.
So we will know.
Yeah.
And the other thing, do you think it would be useful to extend this to polymerase register, or the raw data at this point to investigate?
So polyploid depends
Means
like
only
how
like-
much crazy like if we want to go to 12 plotty, I don't know. I have like
Mechanics too.
in practice probably. And also it requires more sequencing data. We generally do like 15x per haplotype. So even that there is limited.
So I think haplotyping on cancer data would be more complicated because of this
Yeah.
explosion of. Okay, other questions? Any observation of the open system?
Maybe another 10, 15.
Okay, thank you. So you totally like the coffee break. I don't think you're still ready. Thank you.
Thank you.
Almost linear.
Yeah, okay. So right now in that part,
Yang